Today I want to just talk about biofilm modeling, nanomaterial dynamics, but also some of the work that I've been doing. I'm currently on junior leave, so this weird sabbatical-ish thing that Duke does right after our mid-tenure review, where I got the opportunity to work in Romnick Xavier's lab at the Broad Institute, trying to model human microbiome and immune system interaction, really trying to learn a little bit more about the immune system so that I can at least understand what people are talking about. I won't ever understand all of immunology, but I'll at least understand what they're talking about when they talk about CD4 plus cells and CD8s and all the rest of that stuff. But first off, I want to just ask a question, maybe trigger some nostalgia. Does anybody remember their first science experiment? I mean, I could ask about, like, I don't know, your first derivation. You might remember that one, but then it's probably going to be like, I don't know, most of us are going to remember deriving, I don't know, the quadratic formula or something like that. I remember my first derivation that I really remember was like, I was sitting at the kitchen counter. I was 15 and I said I was bored. My dad, who was a math professor at PUI, said, you know what? Like, derive the integrating factor for first order homogeneous equation. I was like, okay, fine. But like, I felt really proud at the end of it. But no, anybody remember their first science experiment? No?
Yeah.
Electrical outlets count.
There we go. Yeah, that counts. Sticking stuff in electrical outlet. Let's just kind of figuring out, hey, how do electrons transfer? Mine, I remember my first science fair project because it was first grade, and what I did was I kind of looked at my mom's coffee cup and the coffee mold that was occasionally growing inside of that and said, you know what? This looks really cool, and I want to do this for my first science fair project where we compared mold growing inside of a coffee cup versus mold growing inside of like coffee grounds. That was like my first introduction to scientific experiments. And it turns out that in microbiology, that's kind of the first thing that we start teaching students when identifying fungus is, well, how to identify bacteria or fungus based off of their shape. So first grade me had some interesting foresight, maybe. What I'm talking about here, though, are biofilms. So the image that everybody's going to have in their head of bacteria when they think of it, they're going to think of, I don't know, potentially random things swimming around inside of a glass of water that you pulled out of, I don't know, say Washington, D.C. right now. Right? Like, that's the image in your head. And that image is wrong, though. The image that where bacteria like to live stuck to stuff. They like to either live stuck to each other or stuck to, like this right here is a piece of iron oxide in river sediment. And this right here, the famous Yellowstone mats, right? These are bacteria, largely giant collections of bacteria and algae. This is a bacteria that you should all be familiar with. This is why we have to brush our teeth every day, right? We brush our teeth twice a day in order to cut down on the biofilm that's developing on our teeth. You're never going to kill all your bacteria. That would actually probably be really bad. If you killed all the bacteria in your mouth, they help you digest some of the starches that are in your mouth. So don't do that. But you at least want to cut down on that biofilm layer. This is a biofilm I studied during my PhD, which basically took that little bottom organism and I put it in a nice controlled reactor so I could see impacts of shear stress on bacteria metabolism and bacteria structure. That's really what biofilms are, right? That's really where bacteria like to live. The bacterial life cycle, the planktonic stuff that we see when we think, oh, those little things swimming around in water, that's only like 20% of their life cycle, right? Most of the time, what they're doing is they're sticking to each other. They're sticking to themselves. They're sticking to a surface. They're secreting a bunch of different polymers so they can protect themselves from predation, protect themselves from attack from other organisms, and also make sure that whatever nutrient source that they've found, they can keep. Normally, what you'll find is biofilms are generally of a single lineage. So one organism attaches, and then the rest of them just start, as they divide, they're like, these are all my kin. These are all the people that I like hanging around. Everybody else stay out. That's generally the way biofilms like to be organized until you start talking about multi-species biofilms, and that gets complicated. Then they'll disaggregate. They'll run out of nutrients. They'll send some quorum sensing signals, and they'll all leave. All right, and that's when we start seeing those little free-floating things swimming around. This is what my lab likes to study. So we study biofilm physical chemical dynamics. We also try to do modeling and scaling analysis to really make our work kind of universal and universally applicable. We design new experimental tools and methods to really study and identify what's going on inside of a biofilm. And we also try to apply some of this to problems and challenges in water, wastewater engineering, as well as floods. So the bottom left image is an image of after Hurricane Helene hit Western North Carolina. about 2 years ago now, close to 2 years ago. It'll be 2 years in September. We went out and we sampled fungus from flooded out buildings. And we, again, did that lovely morphological identification and said, hey, what are the different isolates that we can get? Then we did some, ITS sequencing to identify the different fungus that were growing there, hoping to help people recover from floods. So that's my lab in a nutshell. But this was not the path. So for any postdocs in the room or PhD students in the room, this is not how I got there. Right? I did not do that. I did random stuff. I wanted to be an intelligence analyst briefly. I wanted to do materials engineering for jet design. Let's see what else. I was tempted to be a short order cook, right? Going through graduate school, typically the alternative career is baking, right? So I catered for like 500 people once. It was awesome. But yeah, didn't do that. Did some other crazy things. Yeah. So that was me. Not a linear path at all. Quick disclaimer, Duke submitted patent application for some of the technology that's going to be discussed in this presentation. So just that. Another question I want to ask everybody before we really start getting into what I've done in the last, me and my lab have done, is I want to talk about what we've done as society. We've improved life expectancy. Anybody want to guess as to what this inflection point is right here? What do we do?
Hygiene.
Hygiene, yes. Most people will say antibiotics. You said hygiene. That's like the first time I've gotten that as the first answer. It is hygiene. It is washing your hands, right? Washing your hands means that, I don't know, if you're performing a C-section, the mother's not going to die in childbirth, right? If you are digging a bullet out of a president that just got shot, the president's not going to die because of sepsis, right? Like those things are not going to happen, right? So we're going to increase people's long-term health. This is when antibiotics were introduced. Antibiotics, when we introduced, let's say, penicillin back in 1945, before we introduced it, we actually knew about antibiotic resistance for penicillin. When you look at a broad spectrum of antibiotics that have been produced, you'll see that most of the time we have about like 30-ish years before we find antibiotic resistance. Linezolid is another one where we knew about resistance before we started introducing it. Vancomycin has probably been one of the longest lasting ones. That's why we also try to use it as like the antibiotic of last resort, because it took us a while to find antimicrobial resistance to that one. Antibiotic development and approval has really been decreasing over time, because a lot of the antibiotics that we use are things that we're using from bacteria microbe warfare or bacteria warfare. And if you're thinking about fungus in particular, if you're looking at fungus warfare and trying to derive an antibiotic from there, our cells look very similar to fungi, right? So it's generally things are going to be somewhat toxic to us. Translational research should help, right? So interacting on a level where you're talking to policymakers, where you're talking to engineers, when you're talking to doctors, even when you're talking to patients, that should help reduce the amount of antimicrobial resistance, right? So basically telling people you have the flu, you don't need an antibiotic, like that should help. But then we as engineers and scientists, one of the things that we're going to try to do is we're still going to try to develop new tools, right? So we're still going to say, you know what, nanoparticles that were seen by investors, policymakers, and engineers is a really easy to design and engineer tool that bacteria really shouldn't be able to develop resistance to. We thought, you know what, there might be reactive oxygen species that are going to kill the bacteria because some of these nanomaterials are going to be derived from catalyst. Turns out you don't really get that inside of the cell because there's too many things that might coat that material and just the reactions aren't in the right conditions to produce reactive oxygen species. You might have membrane wall damage that might start penetrating the cell. Turns out bacteria cells are a little bit tougher than that. You hopefully might have some protein damage and some DNA damage. Those bacteria can start evolving resistance to. In fact, this right here is E. coli, like lab grain. You can drink it, kind of safe E. coli. Don't do it, but you can. Like K12 will not make you sick. And what it's doing is it's secreting flagellin. So that's part of the thing that makes its flagellin motor protein into the surrounding environment and agglomerating silver nanoparticles, right? Rendering those things that might be in your clothes right now completely useless. When I say engineers tried this, there's me. Right there, I was an engineer looking at aloe vera-mediated tellurium nanoparticles and saying, you know what, this should hopefully cut down on antimicrobial resistance. So I wound up quantifying the carrying capacity, the growth rate, and the lag time. So carrying capacity is in A, lag time is lambda, growth rate is mu for this model down here, and say, you know what, can we predict whether or not these bacteria are going to develop resistance? With growth rate, there is no dependence on concentration. There is dependence on concentration. With carrying capacity, there's no dependence on lag time, there's no dependence on concentration. And with carrying capacity, there's no dependence on concentration for this newly derived alveoli-mediated tellurium nanoparticle. Concentration dependence is one of those things where we can say, okay, bacteria shouldn't be able to develop resistance if there's a concentration dependence on these two parameters. Turns out that back in 1975, someone had discovered a handful of conserved genes in multiple species that would produce tellurium resistance. So bacteria have seen this lovely toxic metal before, even for nanoparticle size tellurium. And that's because when we think about bacteria, they've been around for a couple billion years, right? Old Earth interactions with metals that are completely sequestered that we couldn't survive around, but they had to figure out a way. So nanomaterials is kind of the summary of that. They haven't really been used that often, right? So if you look at nanomaterials as implanted medical devices, they really aren't translated at a high rate, even if you have a high publication rate, which, I mean, that's common for most things, but for nanomaterials, it just really wasn't there. Antimicrobial peptides are currently all the rage. Everybody looks at an antimicrobial peptide and says, well, we produce them to kill off our bacteria and to kind of maintain this homeostasis. And they shouldn't really be able to develop resistance against. We should be able to apply machine learning to design new drugs that will take them out. Turns out that's kind of hard too, because bacteria have developed a handful of resistance mechanisms because, again, we use them, right? If we produce antimicrobial peptides, the bacteria that are going to exist and try to attack us will have developed resistance to them. What about biofilms, right? So I started out this talking about biofilms. So biofilms generally make the problem worse. So over here, what you're seeing on the left-hand side are biofilms produced or growing on implanted medical devices in a person. And one of the things that happens after, let's say you have a coronary artery bypass graft, so the CABG, they might try some antibiotics. So you might try daptomycin and naflocin, but at the end of the day, what you're going to have to do is go in, scrape out the biofilm. So literally open up the wound again, scrape out that biofilm, then treat with antibiotics because biofilms are somewhat difficult to treat. Biofilms cause more cell death than your normal bacteria. So your normal than your planktonic bacteria. I spent all that time explaining that planktonic bacteria is just like 20% of their life cycle. And then I made that mistake. But yeah, so this right here is a tunnel assay. The tunnel assay, what it shows is how far along your cells are toward dying, right? So the increasing probability that your mammalian cells might die. And the higher that is, well, the closer to death those mammalian cells are. And what you see here is the planktonic cell media, so media from surrounding planktonic growing bacteria, versus biofilm cell media shown in a lighter gray. Let me use a laser pointer for that. There we go. Okay, so biofilm cell media is in this light gray, planktonic cell media is in a dark gray, and what you're seeing here is that there is an increasing probability of death with biofilm cell media, right? Biofilms are resistant without really developing resistance. So if you look here, you can see that biofilms are able to withstand higher doses of ciprofloxacin than planktonic cells. However, your biofilms actually, even after you've passaged them like 6 times, so you've kept growing them up in a small amount of ciprofloxacin, theoretically killing off some, trying to evolve a new lineage that might be more resistant, they don't change their minimum inhibitory concentration, right? They never develop resistance. Whereas your planktonic bacteria, they do. They start saying, you know what, I can survive more and more of this antibiotic. You have to use more of that antibiotic to try to kill off this biofilm, to try to kill off the planktonic species. And that's because biofilms are really recalcitrant. They are not resistant. That is, they have these different polymers that prevent antibiotics from penetrating into the biofilm and actually getting at the cell. All right, so they'll secrete things like extracellular DNA, or as I mentioned earlier, flagellin, out into the media that will trap your antibiotic and make it stuck so that the bacteria never have to see it. Bacterial recalcitrance is theoretically a function of periphery sequestration. So this lovely experiment that I'm going to talk a lot more about was done by Zhang et al. What they did was they grew spherical biofilms, and they treated those spherical biofilms with fluorescently tagged tobramycin and ciprofloxacin. They also tracked cell death, and they said, okay, who's alive and who's dead within this species? So they were tracking this over time, and they were also tracking who was dying and who was living. The reason why they modified charge is because it was previously thought that positively charged nanomaterials would penetrate better into a negatively charged biofilm space, right? Think charge-charge interactions. However, this study by Catherine Murphy down at UIUC, or at the time she was at University of South Carolina, but she's moved to become chair of UIUC's chemistry department. This study showed that, you know what, actually negatively charged nanomaterials are going to be absorbed inside of a biofilm, right? In an estuary biofilm, so like something that you might see on a river. No metric really seemed to capture nanoparticle accumulation in multi-species biofilms. So here you'll see the charge, whether the negatively charged or positively charged, shows similar bioconcentration factors. Here you see that hydrophobicity, it's like a more hydrophobic and less hydrophobic, doesn't really describe the amount of biofilm accumulation of a nanomaterial. And right here, you see that you can use the same nanomaterial, so same charge, same type, and get two different responses, right? So what's really going on is the question that my lab tried to address. And that's because on a impact level, on a social level, there's really no discernible impact of bacterial of coating, let's say, these vascular grafts. When I said, yeah, the implanted medical devices, biofilms are a really big problem inside of there. There seemed to be no real discernible difference there, right? We are not able to prevent biofilms from forming on a vascular graft, whether we coat it with nanomaterials or antibiotics, right? You're still going to have biofilms forming on side of these vascular grafts or implanted medical devices. So what do we do, right? So this right here, this is one of my kids' favorite stories, favorite characters. This guy named Pete the Cat. He is walking along, wearing white shoes, singing a song about how much he loves his white shoes, and then he winds up stepping in a pile of red strawberries and turns his shoes red. And he's like, does he cry? No. He just keeps on walking, right? He keeps on saying, you know what, we can keep going. And that's kind of where I think that we should all go as a field, right? Despite all the doom and gloom that I just said about we can't develop new antibiotics, antibiotics don't seem to work on vascular grafts, that charge doesn't seem to be a factor, we should still be able to go forward, right? So the first thing that my lab did is we looked and said, okay, how do we model charge effects on uptake? So we took the diffusion equation, so Fick's law. It's light, it's really bad. There it is. So we took the diffusion equation, so dCdm is equal to change in diffusivity times change in concentration, right? So we took that, then we added on the Nernst-Planck equation to that and said, okay, how does charge impact mobility of a species based off of both its permittivity in free space, the zeta potential of that particle, and the electric field that the surrounding media is going to produce. And we also added in binding kinetics. So that is how fast your antibiotic could get stuck to a part of the biofilm and then how fast it could leave. So this Kf for being stuck to the biofilm versus Kr, which is that nanomaterial or antibiotic being released from the biofilm. So we did that. We did the thing that all good research should do, which is non-dimensionalize the equation. So non-dimensionalizing things that popped out, we popped out a dimensionless diffusivity constant along with a porosity. We also popped out a kind of a dimensionless version of potential and a dimensionless forward and backwards reaction rate. So we did that. And what we found was that charge really did not appear to matter. So we were able to find two different pieces of data and said, well, what happens if we add in that electrophoretic term, right? So that Nernst-Planck term that I just mentioned. Well, turns out we get a little bit better fitting. But then when we keep it in here versus not keeping it in here, we get mixed results on whether or not it actually fits the data. So that was kind of a first step saying, well, maybe it's not the Nernst-Planck equation, right? Maybe that charge isn't the thing that we should be looking at. So punchline to this story, what we found is that heterogeneous porosity and heterogeneous attachment site density really seem to describe antibiotic penetration. So ditching the Nernst-Planck term, ditching charge, and really sticking to porous architecture, that's that term in Phi. And a heterogeneous porous architecture as well as a heterogeneous attachment site density. So that's where those antibiotics can stick along the biofilm, has a spatial dependence to it. We were able to fit that to data, that data that I mentioned earlier from Bu Zhang's work, where they were tracking tobramycin and ciprofloxacin into Pseudomonas aeruginosa biofilms, and we got our data to fit pretty well. How we derived that, so where we started was that diffusion reaction equation. So again, looking at a simple diffusivity minus an on and off rate for the antibiotics sticking inside the biofilm. We again non-dimensionalized this. We popped out numbers that we thought should matter. So a Dom-Kohler number, which is a binding rate to a diffusion rate, dimensionless binding constant, as well as an effective diffusivity, things that you can know and measure, as well as a porosity. Porosity doesn't come into this equation. Diffusion reaction equation with heterogeneous porosity, that's where we added in porosity. So we looked at what's called the fixed Jacobs diffusion equation. Fixed Jacobs equation just adds in this area dependence term on how fast your antibiotic can move through a space. So if you're tracking something going through space, you might be, depending on your area profile, you might squeeze tighter and tighter so the antibiotic can't pass through, or it might stay wide and then your antibiotic can pass all the way through. It modifies that diffusivity parameter by taking out the area and the spatial dependence on the diffusion parameter, making it kind of an explicit version. In porosity terms or dimensionless terms, you can get porosity in there. Then we said, okay, well, we have the diffusion reaction equation. We have a heterogeneous porosity equation. What if we add in heterogeneous attachment sites and then combine both that heterogeneous attachment sites as well as the heterogeneous porosity and try to test all four of those models to determine whether or not we can get a model that predicts the penetration of both ciprofloxacin and tobramycin into a Pseudomonas aeruginosa biofilm. First thing we also had to do before we could do that is we had to come up with some sort of term for phi of x and cs of x, because looking at those two terms, looking at the porosity and the binding site distribution, we could try to fit those, but then that would be fitting like 4 different parameters. So we said, let's get some functional form for porosity and some functional form for the binding site distribution. So exponential porosity is not a bad assumption in a biofilm. So this is work done in 2000 showing the porosity in a biofilm. It's somewhat exponential. But when I say not a bad assumption, this right here is work done in 2012, so about 10 years later, and it showed that you have more of a Poisson distribution of porosity in your biofilm. So not really bad, but also not great. Then we, as I said, we used Bu Zhang's data where she had plotted both depth from the surface as a function of time for ciprofloxacin and tobramycin going into your biofilm. Now, the homogeneous model doesn't really capture penetration at all, right? So data versus our model fit in solid. That's that homogeneous model. Your Akachi information criteria and your residual sum of squares, just horrible. Forest order reaction kinetics is useful. It's used, and that's because when you talk about the bulk, right? When you're talking about the biofilm as a whole, not biofilm over space, you can use that, right? So you can quantify how much an antibiotic or a nanomaterial is going to accumulate in the total biofilm using first order reaction kinetics. So I wouldn't completely knock that. It's not useful for trying to predict spatial dependence. The heterogeneous porosity model doesn't really do much better. So just saying, hey, there's a spatial dependence on your porosity, you don't get either tobramycin or ciprofloxacin improvement fits. Heterogeneous attachment does a lot better, specifically for tobramycin. So if you look here, tobramycin is in red, ciprofloxacin is in green for both your Akachi information criteria and your residual sum of squared. And part of the reason for that is that Pseudomonas aeruginosa has these known binding sites for tobramycin. So there are specific sites that your antibiotic is going to want to attach to that Pseudomonas secretes to make sure that it can be resistant against tobramycin. It doesn't have the same thing for ciprofloxacin. So when we added in this heterogeneous porosity, that's when we were able to both drag the residual sum of squares and the Akachi information criteria down for both ciprofloxacin and tobramycin penetration into a Pseudomonas aeruginosa biofilm. In total, this was our model comparison. as we wound up building up more and more complexity into that model, trying to make sure that we didn't add too many terms, that we were not overfitting our problem, and we were describing our problem as simply as we could. The model really can predict periphery sequestration if we look at these two terms that we derived here. So an attachment site heterogeneity constant. So that's just how heterogeneous this distribution of things that your antibiotic can stick to, versus an adjusted porosity heterogeneity constant. So that's how porous your biofilm is, and not just how porous it is, but how that porosity is distributed, whether you have like super porous near the top and then not that porous in the middle, or super porous in the middle and not that porous around the top. Depending on that distribution, you can get complete antibiotic penetration or penetration just on the sides, which is what we see for recalcitrant antibiotic penetration. That's if you know beta and C. So I have some experimental work that I would talk about in a different seminar, where we've started trying to create new devices that we can measure beta and C, so that we can hopefully start doing this for more than just those two antibiotics against that one bacterial species. So right now I'm going to pivot to a different talk. So as I said, I've been on junior leave trying to look at microbiome immune system interaction. And the first question that people should ask is like, why is a biofilm researcher interested in the gut? Right. Clinically, this is what a biofilm looks like in your gut. So before somebody says, hey, you have a biofilm growing inside your gut, we need to treat this with antibiotics, et cetera, they're going to look for that, right? That's what that film that they're seeing, what a healthy non-biofilmed intestine should look like, is just the pink matter, not the pink and brown and speckled matter, right? That's your healthy gut. However, I just told you here that 90% of all bacterial life cycle is in a biofilm, whether you're clinically presenting as sick or not. Most of that life cycle is going to be in a biofilm. So we haven't necessarily discovered what is a good metric for describing a biofilm other than clinical presentation in your gut, right? I think me and other biofilm researchers think that we just need a better biomarker to describe what a biofilm is, and not just limit it to, and that's why that's why this paper exists, is because previously people said, you know what, you have to have a surface or an interface to have a biofilm. But people have known since at least the 80s that pellicle biofilms existed. So biofilms that were just kind of an agglomeration of floating bacteria inside of either wastewater treatments systems or in rivers. So that's why I really wanted to look at biofilms inside of the human gut. The other reason is that health is regulated through this feedback control system of self versus non-self. Right? So this combination of external stress, external neurochemicals, all surrounding your body, plus how much you're sleeping, et cetera, really determines your overall health, right? It's not just, oh, are you infected or not, right? Not just you have an immune system that's strong enough to fight off things. There's also interactions with your microbiota, all right? There's this feedback between self and non-self. So in a disease state, of course, we know that you're going to mount an immune response. But in a non-disease state, if you don't have the right bacterial population inside of you, you may wind up with autoimmune diseases, right? So people have done studies on mice that have no bacteria in them whatsoever, and found that they are less responsive and less healthy than mice that have bacteria, right? So your no-biotic mouse versus your biotic mouse. Health is not as simple as diversity is good. So that was one thing that people were thinking, is that dysbiosis is when your bacteria start coalescing around a single species, right? So let's say that you have a C. difficile infection. So that's one of the things that causes diarrhea. If you have that, that's because that population of that one species of bacteria starts dominating all the rest of the bacteria in your gut, right? Not necessarily the case. So this was a study on children in Finland, Russia, and Estonia in the Xavier lab that I'm working in. And what they showed was that over time, the kids that were born in Russia had very little species diversity. They all eventually coalesced around the same species diversity, so this Shannon diversity index in their gut. But the Russian kids had fewer incidences of autoimmune diseases, specifically type 1 diabetes, egg allergies, and milk allergies, because they started out with a very low diverse population of bacteria in their gut. They were eventually able to identify at least one bacterium that they said, when this dominates, you aren't going to have these autoimmune diseases. From an engineering standpoint and kind of mathematical standpoint as well, the gut microbiome seems to have this fixed set point depending on the person. So there were two people that did this study. So both Lawrence David and Eric Alm tracked their entire gut microbiome for a year. And over time, they both got sick, but eventually their populations wound up coming back to that fixed set point, relatively fixed set point that they had established, right? Whether they were living abroad or not, they kind of came back to the same point. So from a modeling standpoint, I'm like, okay, this is intriguing. This is something that I could aim towards. The current frameworks, though, for understanding immune microbiome interaction, it really focuses on leveraging big data. So what they'll do is they'll take human studies, they'll coalesce these and through multi-omics, they'll generate hypotheses. They might test them either in vivo or in vitro, and then kind of go back around there. They're not really looking at developing mechanistic modeling. My hope is that if we can get some in silico models, right? So if we can add in these in silico mechanistic models, that we can start testing these perturbations and trying to figure out how that really impacts health. All right, so some of the existing work that people have done for this, they've looked at quantitative pharmacological analysis to do feedback control on what happens if you have, let's say, an environmental pathogen that's trying to attack your skin, or let's say you have psoriasis. You're going to treat it with these certain drugs that might knock out some of these immune pathways, so IL-22, IL-17, et cetera, that cause inflammation, that weaken your skin barrier integrity and make it easier for bacteria to get in. And they've done this really through just modeling the immune system and thinking about the pathogens and the microbiome as just this like giant lump parameter system. So that's shown in P. Classic population dynamics, on the other hand, says, you know what, we have this tool, right? We have this tool that a lot of people use. There's some really good work done out of a lab at MIT that I just saw a talk on 2 weeks ago, where they've used Lagavultera, multi-species Lagavultera modeling to show as you add in a new species or a new pathogen, how well does that population respond and how well can it keep those bacteria out? All right. What one person did with this, where they started trying to look at the immune system as well and coupling with this Lagavultera modeling, is they said, okay, we have our population of bifidobacterium, which is one thing that we get from our mother's milk. We have a population of bifidobacterium that we start accumulating just as children as they're growing older. And then we also have the rest of our gut, right? So they term those as X1, 2, and 3. They also said we have this feeding source, so that is how much you're going to get as you're being weaned off of breast milk, whether you're breastfed or not, how that might impact your bifidobacterium population. They also said what happens if we add in an immune system to this. So what happens if we add in both what you can get from your mother as well as what you can get from yourself. So what immune signals you might get from that, and how well does that control your bifidobacterium population? And they always say that with a reasonable guess that this did describe our adult bifidobacterium population. So I thought, okay, well, as a naive, I've never studied the immune system before, that I could create this adaptive feedback control diagram of the immune response and then model that, where I'd say, okay, we have T cells, B cells, lymph removal of those things. So some sort of, you're sucking them away at some rate QC. You'll also have your antigens, which are eventually going to come from your bacteria, and they're going to affect your sensor cells, your epithelium, your macrophages, your dendritic cells, which will signal your lymphocytes, which might change your adaptive immune response. I did this, and then I was told that, I did this because when you read the literature, one of the things you see is there's a whole lot of focus on control in disease states, right? And so subsequently, in a disease state, you are going to have those really conventional cells. You're going to have T cells, you're going to have B cells, you're going to have macrophages, the things that we think engulf cells and kill them off. Turns out that's not the way our gut normally responds. Our gut normally will not have T cells or B cells inside of it, right? They're normally sequestered here and here, right? So inside of these little crypts within, or outside of the crypt, before you get to the luminal layer of your gut, right? So they're sitting here. The luminal layer where all your bacteria live is out here. They don't really like crossing that barrier unless it's disrupted, right? Unless you have some sort of inflammation which allows them to pass through. That's the only reason why they'll get there. All right. Instead, what happens is you might have defensins. So you might have things like IL-23 and IL-1 beta, which might affect your bacteria. but they seem to be species non-specific. So it doesn't seem like there's a good way of capturing the set point that our bodies seem to have, right? Because if you're killing off all the bacteria, so if these defensins, mucins, IgA, and IgG are really just pushing all the bacteria way over here, trying to keep them away from your epithelium, that's not really a good way of controlling your bacteria. So I went and said, I still can think about two different ways that our bodies can control microbiota. All right, so this idea of antigens binding or antibodies binding to antigens and trying to neutralize the bacterium that way, as well as specific defensins that might kill off your bacteria, specific peptides that might kill off a specific species of bacteria. And I modeled that using a Michaelis-Menten kinetics. So that's that term. This is really bad. So let me just use my mouse. So I modeled that using this term m.n. So Michaelis-Menten kinetics describes a binding event, so a two-step binding event of, let's say, a antigen, where it's going to stick to something and it might come off, but most of the time you're going to try to get it all the way on, and it's going to neutralize or create a new product at the end of it. I also just model it using a simple, you can have more abundance, A, of that antigen or that antibody, and then minus an inhibitory effect, right? So I is certain bacteria will produce things that might inhibit the amount of antibodies that the body's going to produce. I combine that with the standard Michaelis-Menten kinetics here. This is what that equation looks like in a kind of a more expanded form. Again, looking at that competitive and non-competitive interaction. I went through the literature and identified some species and some phyla, mostly relying on my colleagues at the Xavier lab, to try to identify what species I should be caring about in the gut. And then one of the things that I did was I had ChatGPT give me a whole bunch of parameters that should hopefully fit these pieces of data. So both mu, alpha, and D were all derived from me just saying, hey, give me the best guess, because I just need to test something right now. And one of the things I found is that on a sanity check, I do get a stable population, which is good. All right, so just straight log of Volterra gives me a sanity, kind of a stable population. I have a nice stable fixed point of when you plot a phase plot of the change in some of the change in population versus just the sum of the population, you have a nice fixed point there, so I can work with that. And I'm like, okay, this is it at least works. On the other hand, when I add in this immune control, things get a little hairy. So one, you'll see over here that I killed off most of my bacteria, and I'm left with a bunch of proteobacteria and firmicutes. You really don't want proteobacteria in your gut. You don't want a lot of them. Right? Proteobacteria, things like E. coli. Right? So you don't want those. And that wound up dominating. Wound up killing off things that we know are in our body. So one, ChatGPT's parameters might just be wrong. Like that's possible. But two, I don't think I have the best control because it worked for the previous model, right? And it's unstable-ish, right? So here's those two phase plots. So instead of just plotting the phase plot for my bacterial population, I also added in a phase plot for my immune signaling. And here you can find somewhat of a stable point, but it's not the best. Preliminary simulations when I modify V with this simpler model. So instead of using Michaelis-Menten kinetics, I'm using just this A minus I. So your thing that's going to kill your bacteria minus the thing that bacteria are producing to prevent themselves from being killed. And there's a fixed stable point that you can find here for antibody population versus bacterial population. If you wind up even looking at your stable points for your antibodies and your bacteria, you do still see those fixed stable points, but it's not the best.
I have a question.
Yep, go for it.
You're in a pretty high dimensional parameter space, and you have some parameters, and maybe it can do the thing you want it to do. I guess that's maybe not super surprising given how many parameters you have. So what do you have to actually constrain the model behavior?
So hopefully what's going to constrain the model behavior is that Michaelis-Menten term. But you're right that with the parameter space of having, let's say 34 different immune signals and 34 different bacterial species and 20 different immune signals. Yeah, that's a pretty high dimensional space. What can constrain it beyond that, I'm actually not sure. That's hopefully part of the reason why I'm here, is to talk to more people that might have some more insight into how I could potentially constrain this problem. Yeah, so that's where I am right now. There's a little bit more stability when I, of course, decrease V, but what I've also mentioned here is I have these other four parameters. So this K parameter, both an inhibitory and a competitive and a non-competitive K. And it's like with those being set to 1, trying to just say, you know what, maybe I can ignore those terms. Maybe that wasn't the best idea. And while right now I'm able to get some model convergence, we still have a pretty long way to go to minimize both this proteobacteria behavior as well as to drag that Shannon diversity index up to where it should be. Right, so it should be somewhere hovering between 4 and 5 if I have the right immune signaling and the right microbiome signaling, but right now I'm nowhere near there. Long-term goal is to get that fixed set point defined by H prime, to define immunological microbiome responses using basic linear control theory building blocks so that hopefully people can start using this model and building more and more complex ones, and then potentially doing dynamic set point / adaptive control theory because our bodies operate with an adaptive immune system or immune response in addition to an innate immune response. So for the first half of the talk, I've had to acknowledge my graduate student, Josh Prince. He graduated in 2024. He was a 2024 President Management Fellow. He's currently at ICF. And then I also have to acknowledge the Xavier Lab, which has started teaching me about immunology. I should also acknowledge the NITMB for this too. And thank you. Any other questions? That was a great question, though. Yeah.
How much does surface tension affect the ability of a biofilm to stick to its surface? Because I know people have been working on like these anti-wetting surface.
Yeah, so the question was, how much does surface tension affect biofilms either forming their own structure or potentially from sticking to a surface? And one, I guess I showed that data on hydrophobicity versus hydrophilicity that people were thinking, good. Maybe if we make a super hydrophobic surface, that's going to prevent bacteria from really being able to stick to a surface. And it turns out that I think it was E. coli. Somebody showed E. coli just bouncing up and down on this interface between the material, the super hydrophobic material, the water and the air layer, so that it eventually pushed that water layer. It increased the energy of the water layer so that it broke that air gap and the surface got completely wetted again, which was crazy to see. It's a really awesome video to watch. It found that out in early 2000s. So surface tension as far as or hydrophobicity as far as a way of preventing infection on implanted medical devices doesn't seem to be that fruitful. As far as defining how the shape of your biofilm, their surface tension hopefully potentially plays a decent role. I know that Jorn Dunkel's group showed a lot of really cool phenomena of bacteria under high density secreting different surfactants and changing their shape, right? And changing how fast they moved. So there's two different stories there. One is, can we use that as an infection prevention? Probably not. But two, they do it, and that might control some of their morphology.
Yeah. In your first story where you were fitting the model,
yep.
How well constrained are the parameters in that model? The heterogeneous porosity like that.
Let's see. Nope. And I stopped sharing. Let me reshare. Oops. Share that. So the question was, how well constrained are these parameters? And the answer to that is, I don't know. We didn't test it beyond trying to get both convergence and fits to the existing data. So that's where we left this problem. But no, that'd be an interesting thing to look at. I know right now the next goal out of that was actually to get a 3D model, and so that's a paper we're trying to write up right now. It wasn't to kind of test the rest of the model behavior. It was like, okay, we have this working in 1D, but of course all biofilms are 3D. The beautiful thing about the data that we were fitting to is that it was spherical, so we could do that axial symmetry, but that's to grow that biofilm in a spherical shape is hard and just kind of it's hard. And so we were like, can we get the common biofilms that people work with?
I've seen claims, I don't know if they're true or not, that some people say that certain types of metals are more antibacterial than others. Is that true, or is that a fallacy?
That's true to a certain extent. Like there have been enough studies showing that copper does produce, I want to say superoxides. Copper has some cellular damage, and I forgot why. Yeah, I'm blanking on why copper works. Gold doesn't work, even though it's a great catalyst. Silver, I showed you that example of silver not working. One of the things with being antimicrobial, especially for implanted medical devices, is that once you put it in the body, it's no longer pristine. So we want our body to coat whatever we put inside of it with something that it can start recognizing, right? Because otherwise you're going to have inflammation, you're going to have your body rejecting that device. And so you can coat it with the most pristine nanotextured surface, but the first thing your body does is it's going to coat it with proteins. And then the bacteria are going to stick to those proteins. And then you're stuck. There's another large problem with biofouling on ships. And so with biofouling on ships, what happens is you can start out with your pristine surface. First thing that'll stick to it are going to be bacteria. And then you're going to get those things that, like the mollusk and the other shell things that are going to stick to your ship. So again, you can keep this pristine surface, but then bacteria foul it, and then you're stuck. And you had a question back there?
I was curious, you talked about the gut with the biofilm.
Yep.
And the healthy gut and the non-healthy gut, but
Yeah.
some biofilms are not necessarily bad on the.
Yes.
So I wondered also with the implants, are all biofilms that grow on the implants necessarily bad?
That's an interesting question that I had. So the question was, are all biofilms that grow on implants bad? And I mean, because I just said that, no, yeah, in the gut, you still want bacteria. In your mouth, you still want bacteria. Even in your lungs, you have bacteria, right? You normally don't want a bunch of them, but you still have to have them. And so that's a question that I'm not sure if the literature's really investigated a lot, because most of the time when you're pulling out a implanted medical device, you're pulling it out because it's caused inflammation and it's caused some damage, which is the same problem here, right? So the only time that we're looking inside the gut is when there's a problem, right? And the only models that we have of our immune system right now interacting with the microbiome is like when there's a problem, and then you start getting all these CD4 plus cells and CDH cells and all the T cells and B cells kind of migrating and doing their thing. Like we had those models because we're looking for what happens when things go wrong. And we aren't necessarily sure about what happens when things are going right. I think actually the Xavier lab that I'm working in right now, when I presented this research to Ramnik, I think it was last week or 2 weeks ago. It was like, okay, yeah, we don't have enough healthy data. So he's actually about to run a pilot study looking at where is that region? Nice region here. This region of the gut, so way away from your sphincter. So this is where everything comes out. But like up here, it's up there in the lower in the small intestine. In the small intestine, we have 3 different regions of the small intestine, and he wants to track both like the first part where you don't have a lot of bacteria and the second part where you do, and try to see what are the immune signals that are happening in there, because that might help give us some sort of a clue as to what a healthy immune response is going to look like in the gut.